Hello students, so today we are going to learn the second part of the chapter reproduction lower and higher plants. Now in this particular slide you can see animation image of the flower. So you can imagine the beauty of the flower of the nature, right? Now again the second slide again added few more beautiful animation images of the flower, how they bloom. Okay, and also the content what you are going to learn in this particular part of the video, that is the sexual reproduction, structure of anther, microsporogenesis, development of male gametophyte, structure of anatropous ovule, megasporogenesis, and development of female gametophyte. Okay. Now, coming to the third slide, the introduction part of the sexual reproduction. Here we can see a beautiful labeled flower. Just can go through the different labelings starting from the pollen grain, stigma, platform for pollen to land on, style supports the stigma, wall of the ovary, cavity of the ovary, nucellus, embryo sac, micropyle, integuments, all these which form the ovary. Then receptacle, the stalk part of it, the placenta, green sepal, filament, then anther site of pollen production, and the petals. Now the introduction point. The flower is specialized reproductive structure of a plant in which sexual reproduction takes place. So as we know, the flower has the androecium and the gynoecium, the male and the female part where the reproduction takes place. Second point, typical flower consists of four different whorls: calyx, corolla, androecium, gynoecium. That is the four parts. Third, sexual reproduction is characterized by fusion of male and female gametes, that is fertilization. So when fertilization occurs, the formation of zygote and followed by embryogenesis, that is formation of embryo takes place. Now in detail about the sexual reproduction. It involves fusion of two compatible gametes or the sex cells, that is the male and the female gamete, which because of the fusion it forms the fertilization and thereby formation of zygote and the embryo. Further, all organisms reach to the maturity in their life before they can reproduce sexually. So initially the flower reaches the maturity, this formation of the male part and the female part, androecium and the gynoecium. In the plants, the end of juvenile or the vegetative phase marks the beginning of the reproductive phase and can be seen easily in the higher plants at the time of flowering. So initially it is a vegetative phase which then ends and begins with the reproductive phase. The flower is a specialized reproductive structure of a plant in which sexual reproduction takes place. So as we just learned in the previous slide, flower is the specialized reproductive structure where the reproduction takes place. The function of the flower is to produce haploid gametes and to ensure that fertilization will take place. So the haploid gametes, it is male gamete and the female gamete, when they fertilize they form the diploid gamete. Typical flower consists of four different whorls, that is calyx, corolla, androecium and gynoecium. As we know, calyx and corolla, they are the accessory part, whereas androecium and gynoecium, the essential parts, right? Further, sexual reproduction involves two major events, that is meiosis and fusion of gametes to form diploid zygote and the production of genetically dissimilar offspring. So here as two are the major event, meiosis, the first one whereby the division takes place, and then fusion of the gametes which forms a diploid zygote and further the embryo. Then variations are useful from the point of view of the survival and the evolution of species over the time. Now as the fertilization occurs, it leads to formation of zygote, then the embryo. Now embryo, they may be genetically dissimilar offspring, means they may differ from the parent, and this actually is a beneficial. Why? Because this leads to variation, and the variation leads to evolution of new species. That is why we have different type of plant species, maybe of the same genus, the species may be different. Further, sexual reproduction is characterized by fusion of male and female gamete, which as we term as fertilization, the formation of zygote, and embryogenesis, that is formation of embryo. Sequential events that occur in sexual reproduction are grouped into three distinct stages, that is pre-fertilization, fertilization, and post-fertilization. That is what happens before the fertilization, what happens during the fertilization, and later on in the post-fertilization. As we know, the pre-fertilization, the different parts or different worlds formation takes place. The vegetative part, during the fertilization the formation of zygote and the formation of embryo takes place, and the post-fertilization as we learn about the formation of seed, fruit, etc. Right? Then the male reproductive wall of flower is called as androecium. So the male part is called as the androecium. The individual membrane of the androecium are called as stamen. Now these stamen individually has filament, connective, and anther. So there are three parts in the stamen. Moving further, the female reproductive system or part. So female reproductive wall of the flower is gynoecium, and here it is otherwise a pistil. The individual membrane of the gynoecium is called carpel, that is megasporophyll. A flower with many free carpels is called as apocarpus. For example, Michelia species. That's why I put the flower image also, so it will be easier to understand the from the image of the flower how the species looks like. Second, a syncarpus. Now the term syn means what? Fused. Syncarpus flower is one that has many carpels fused together. Example, brinjal. Now brinjal, all of you have seen, some of you like very much, some of you don't like very much, right? But if you see the flower, it's very beautiful, right? Then typical carpel has three parts: ovary, style, stigma. So the carpel has got three parts: ovary, style, and stigma. The number of ovules in the ovary varies. That is, it may depend from flower to flower. That is, paddy, wheat, and mango are uniovulate, whereas tomato and lady's finger are multi-ovulate. Now again I put some of the images in the next slide. So you can see we have heard about the plant, but very few might have observed the flower. So that's why I added the image of the flower, which actually the image makes us to understand the concept easier, and also can see the different flowers in nature. The mango flower, all of us like mango, but see the flowers how beautiful they are. A corn flower, or you can say the maize, paddy or the rice flower. See the rice plant, which commonly the paddy seen in the paddy field, but if you see the flowers very small, looks beautiful. The lady's finger, that is otherwise called as bindi. Again you can see the flower how beautiful it is. Then flower of the tomato. Tomato, again if you see, which is commonly used in the food making, but here you can see again how the appearance of the flower of tomato is. Now moving further, structure of anther. So coming to the male part, the wall of the mature anther consists of four layers: epidermis, endodermis, middle layer, tapetum. So there are four parts in the anther. Now again you can see the image over here. First we can go through the image so it will be easier to understand the concept. So here you got a vascular strand towards the center, the outermost layer, that is the epidermis. Then there is a small groove which is a stomium, microspore mother cells toward the center of each of the pollen sac, the middle layers, tapetum. Okay, so here again you can see the section: endothecum inner to the epidermis, the middle layer inner to endothecum, the microspore mother cells towards the central part, and the stomium here. And here you can see endothecum epidermis layer, the pollen grains, stomium. Now whenever the pollen grains mature, they desiccate. You can see how they disperse. Okay, so that I again added few animation images of disperse of the seed, which is very beautiful when you observe it. Clear? Now here you can see the next point. An immature stage of an anther is represented by a group of parenchymatous tissue surrounded by single-layered epidermis. Anther is generally diathecous, having two lobes. So the term diathecous, two-lobe structure, and tetrasporogeate means tetra means what? Four. So you can see 1, 2, 3, 4, right? So again we come across various terms now. So these terminologies are very much important from one word or MCQ point of view. Okay, and understanding the concept is very much important. So each monothecous anther contains two pollen sacs. In diathecous anther has got four pollen sacs, which you can see over here. Now therefore it is tetrasporogeate. That is why there are four sporangious sac present, or you can see sporangious spores over here. The heterogeneity arises when some hypodermal cells get transformed into archisporial cells, or as we use the term later on, megaspore mother cells. Now here we can see how the dispersal of seeds takes place. You can see how beautifully the seeds are dispersed, or it's like bursting and it is released. And again you can second image, you can see how the crackers explode. So this dispersal is somewhat similar to that appearance as it bursts open to release. So there is various modes of dispersal, right? Then you can see the third image where you can see pollen grain which comes from the trees, weeds, grass. So you can see here animation image continuously shifting, where you can see the pollen grains from the grass, trees and weeds. Now moving further, TS of anther. The archisporial cell divides into an inner sporogenous cell and outer primary parietal cell. So you can see the section of the anther. So here the archisporial cells, it divides into inner sporogenous cell and outer primary parietal cell. The sporogenous cell forms sporogenous tissue. Each cell of sporogenous tissue is capable of giving rise to microspore tetrad. Again the term tetrad, 4, 1, 2, 3, 4. So there is 4 microspore tetrad present. Then parietal cell undergoes division to form anther wall layers. So the parietal layer forms the wall layers, as we just read in the previous slide. So just first we go to the image here. Which are the layers? The epidermis, the outermost, endothecum, the inner one, middle layer, inner to the endothecum, tapetum, the innermost part, and inner which is the sporogenous tissue. Also towards the central part of this, you can see the connective tissue. Then further, the wall of the immature anther consists of 4 layers, as we just read out. First, the epidermis. It is the outermost protective layer made up of tubular cells. So you can see here it is continuously covered throughout. Then second, endothecum. It is subepidermal layer made up of radially elongated cells with fibrous thickening. So it is the second layer, inner to epidermis, which has got fibrous thickenings. Inner to endothecum is a middle layer made up of thin walled cells, one or two layered, which may disintegrate in mature anther. So as anther gets mature, it gets disintegrated. Then fourth, tapetum is the innermost nutritive layer of the anther wall. It is immediately encloses the sporogenous tissue, that is the microspore mother cells, or in short MMC. Now coming to microsporogenesis. Now what is microsporogenesis? Microspore, the pollen grain, genesis, formation. So formation of microspore. So again you can see the image first, the animation image of the pollen grain, how it is spiny with the exine outside, and you can see here small pore also, right? What is the spore? The germ pore through which the pollen tube arises out, correct? Then coming to the points, each microspore mother cell divides meiotically to form tetrad of haploid microspores, that is the pollen grain. Now, structure of microspore. The typical pollen grain is a non-motile haploid unicellular body with single nucleus. It is surrounded by two-layered wall called sporoderm. The outer layer, exine, is thick and made up of complex non-biodegradable substance called sporopollenin. So again another important term, sporopollenin, which is present and helps the pollen grain from degrading. Okay, and the layer, two-layered wall, which is the sporoderm. Further, it may be smooth or with a sculptured pattern. So here one which you can see as the exine. There are pollen grains which does not have the exine, which may be smooth. For example, in wind pollinated, you can see moreover smooth pollen grains, and in case of insect pollinated, bird pollinated, is moreover presence of exine. It is resistant to chemicals. At some places, exine is very thin, showing thin areas known as germ pores. So you can very well see the germ pore, and in this image you can see 3 germ pores present. So in this case there are 3 germ pore present. The other parts, the nucleoli, cytoplasm, in time the exine, outer spiny part, and thickening of the cellulose through the inner to intine. Okay, further these are meant for the growth of emerging pollen tube during germination of the pollen grain. The inner wall layer, intine, consists of cellulose and pectin. So intine is made up of cellulose and pectin. Now moving further, how the development of male gametophyte takes place. First, see the image. So your half the learning will be done with the observation of the image only. You can see development of male gametophyte, the pollen grain, the structure just now as we learned. Further, it has got 2 parts. One is a vegetative, other is a generative cell. Then further, germ pore, generative cell. That is the figure number C. Then D, you can see here the germ pore and the generative cell which is started to divide. Then vegetative nucleus, the 2 male gametes are formed, and further through the pollen tube the 2 male gametes and the tube nucleus are carried forward through the style into the ovule, right? Okay, so first point, pollen grain marks the beginning of male gametophyte. So pollen grain itself means the beginning of the male gametophyte. It undergoes first mitotic division to produce bigger naked vegetative cell and small thin walled generative cell. The vegetative cell is rich in food and having irregular shaped nucleus. The generative cell floats in the cytoplasm of vegetative, so it remains floated. The second mitotic division is concerned with generative cell only and gives rise to 2 non-motile male gametes. That's what you can see here, formation of 2 non-motile male gametes. The mitotic division of generative cell takes place either in pollen grain or in the pollen tube. So the pollen tube carries the male gametes. So this formation either it can take inside the pollen grain or in the pollen tube itself. The pollen grains are shed from the anther at the 2 cell stage in most of the angiosperms. In most of the angiosperm, it is shed from the 2 cell stage. Clear? So from the image, it is very clear the stages. I just see the first part A, pollen grain, B, vegetative and generative, C, the germ pore, generative cell starts to divide. Here you can see the division, and here 2 male gametes are formed in E, and in F the male gametes are passed through the pollen tube. Now moving further, structure of Anatropous ovule, coming to the ovule part, that is the female gametophyte. So here again first see the image, then the theory part becomes easier. So if you start from the anterior end, the chalaza, the nucellus, the inner part, integuments, which are, if you see, two layers are there, so it is called as bitegmic. Then embryo sac inside the nucellus, the egg cell which is present inside the embryo sac, the synergies along with the egg cell, that is the egg apparatus, the micropyle, the small opening over here, funicle, the stalk part, hilum, the connectivity, the definite nucleus towards the center of the embryo sac, and antipodal cells at the chalazal end, and raphe, the dark marcation which you can see, which is even present after the seed formation, like you can see the bean seed or rajma, where you can see this raphe. Okay, coming to the points. Each ovule develops inside the ovary and is attached to the placenta by a small stalk called funiculus. You can see over here. So this funicle is the stalk part. Further, the place of attachment of funiculus with the main body of ovule is called hilum. So you can see funicle attached to the main part of the body through a small part called as hilum. Further, in angiosperm, the most common type of ovule is Anatropous, in which micropyle is directed downwards and is present adjacent to the funicles. So here, one important aspect to add over here, which ovule is this? Anatropous. Now there is another type which we call as orthotropous. Now what is that orthotropous, or difference in ortho and ana? Now in its Anatropous ovule, the micropyle and the funicle lies at the same end, whereas the chalaza on the opposite end. Okay, whereas orthotropous ovule, chalaza will be at the down end along with the funiculus, whereas the micropyle will be at the opposite end, at the anterior end. So when the micropyle is at the anterior end, whereas chalaza funicle at the posterior end, then it is called as orthotropous. Now this is the Anatropous position, where chalaza is the anterior end, towards the posterior end the micropyle and the funiculus. Clear? Okay. Then the ovule consists of central parenchymatous tissue, the nucellus, which is surrounded usually by two protective covering called integuments, outer and inner integument. A narrow opening at the apex of the ovule is called as the micropyle, that is this opening, and the two integuments over here. Further, chalaza is the base of the ovule directly opposite to the micropyle. Now one we can see in the anterior end over here, chalaza, which is actually the base of the ovule. Further, embryo sac, that is a female gametophyte, is oval, as you can see in this image, multicellular structure embedded in the nucleus, as it has got mini cells, as we learn further the 8-cell stage, right? Now coming to Megasporogenesis. So again in Megasporogenesis you can see the image. Okay, first we can read the points. It is the process of formation of haploid megaspores from diploid megaspore mother cell, that is MMC. Further, these megaspore mother cells becomes distinguished into the nucellus more or less in the center but towards the micropylar end of the ovule. So here you can see the image now. Now once you understand the image, the further explanation of the development of female gametophyte becomes self-explanatory or easier to understand. Okay, so what is the first point over here, or first figure? Embryo sac, mother cell, which you can see here diploid. Now here by meiosis we can see 4 cells, 4 haploid cells, which is formed, out of which 3 degenerate. Now one of the inner cell, it starts to grow, forms the megaspore of the embryo sac, which is actually haploid. Now this growth is nourished by the nucleus, and as you can see it further undergoes mitosis. Now it undergoes 3 mitotic division. So 1 over here dividing into 2, 2 to 4, 4 to 8. Now this 8-cell stage, they arrange themselves as 3 antipodal cells on the chalaza end, 2 polar nuclei towards the center, and 3 that is the egg apparatus having the egg cell and the synergids towards the micropylar end. So you can see here the mature embryo sac, that is female gametophyte, just before fertilization, 7 nuclei present, 6 are haploid, 1 is diploid. So 1 towards the center, 2 polar nuclei, they are present in the center. That is, it is a cell with the 2 nuclei. Clear? So now learning the points becomes easier. Now we can learn the detailed aspect of development of female gametophyte. So megaspore mother cell undergoes meiosis to form linear tetrad, means 4 of the haploid cells, that is megaspore. Upper 3 megaspore abort and lowest one towards the center of the nucleus remains functional. So as we saw, 3 of them, they degenerate. It acts as the first cell of female gametophyte. Generally, one megaspore towards the center is functional megaspore. It is in fact the first cell of the female gametophyte. It undergoes 3 successive free nuclear mitotic division. So you can see 3 mitotic division taking place. Thus total 8 nuclei are formed, 4 of which are located at each pole. So initially from 1 mitotic division 2 forms, from 2 to 4 and 4 to 8. One nucleus from each pole migrates towards the center and are called polar nuclei, which is present towards the center. 3 nuclei towards the micropolar end constitute the egg apparatus. It consists of large central haploid egg cell and 2 supporting haploid synergids. So in the egg apparatus, one is a female gamete, that is the egg cell, and the 2 synergids, which is at the micropolar end. Synergids show hair-like projection called filiform apparatus, which guide the pollen tube towards the egg. So which helps in the pollen tube to come towards the female gamete. Antipodal cells are the group of 3 cells present at the chelazal end. The 2 haploid polar nuclei of the last central cell fuse to form diploid secondary nucleus or definitive nucleus just prior to fertilization. Thus, this 7-celled and 8-nucleated structure is called as embryo sac. So, as we saw initially, 8 cells are formed, out of which the central polar nuclei is single cell with the 2 nuclei. So, that is why it is 7-cell, 8-nucleated structure. This method of embryo sac development from a single megaspore is described as monosporic development. Monosporic, mono means single megaspore. In angiosperms, the development of female gametophyte is endosporous, endo inside, within the megaspore. Female gametophyte is colorless, endosporic, and is concealed in the ovule enclosed by the ovary. The term concealed, present inside or covered from the external region. Now, here I have used animation image again for making it easier. You can see here how the division from 2 to 4 to 8 takes place, and 2 coming towards the center, that is a polar nuclei. So how a formation of the formation takes place. Again you can see 2 to 4, 4 to 8, and out of the 8, 4 on either side, 1 cell each coming towards the center, forming the polar nuclei, right? And then you can see the synergids and the female gamete, and also the antipodal, the opposite end. Here you can see double fertilization, how the small tube which you can see over here, where the male gamete, which is bluish in color, one along with the female gamete, second along the polar nuclei. So that's how for double fertilization occurs. Then here you can see fertilization in the plants, how the pollen tube passing over here, then releasing the male gametes, where you can see polar nuclei, embryo sac with the male gametes. Again it is 2 dark reddish nuclei, the male gametes, then egg apparatus, antipodal cells. Then the assignment questions based on today's part of the lecture. Clear? I hope it is clear. So thank you all of you. Remaining 2 videos I'll upload it later.